Hello everyone, welcome in this channel. I am Dr. Bile Juice before you, going to talk about regulation of bile acid synthesis. Bile acid is synthesized by liver. It is present in bile. Daily secretion of bile is 500 to 1000 milliliter. This bile consists of composition of bile 97% is water and 3% solids. In solid, organic substances and inorganic substances are present. Organic substances are bile salts, bile acids, lecithin, cholesterol, and bile pigments are present, and electrolytes are present. Sodium, potassium, bicarbonate are present. This is composition of bile. Coming to bile acid is very toxic substance. It is very toxic substance. Therefore, its secretion must be controlled or it is controlled. Very tight control is there to bile acid secretion. First is bile. First is bile. Bile bile. When bile acid is more, its amount increases, then it activates this FXR. That is farneside X receptor. Farneside X receptors activates FXR, that is farneside X receptors in liver. When it is activated, it causes depression of bile acid secretion. Another function of FXR, it stimulates FGF19 in the liver, and this also causes reduction in secretion of bile acid. This is activated formation of FGF19, and that will reduce bile acid secretion. In this way, bile acid is controlled. Now another neuronal control and hormonal control. Neuronal control and hormonal control. Neuronal or nervous control is by vagus nerve. Vagus nerve, when stimulated, causes contraction of gallbladder and relaxation of a sphincter of OD. Contraction of gallbladder causes squeezing of the gallbladder, and biles present in the gallbladder comes to the duodenum. This contracts and this relaxes, then it will come into the bile. Concentrated bile present in the gallbladder is squeezed. It comes into, due to contraction, it comes into the duodenum. Number 2 is hormonal control. In hormones, secretin and cholecystokinin, pancreatinine. Secretin is secreted from S cells present in the duodenum due to presence of acid in duodenum and bile acid. This secretin is stimulated or formed. This secretin will increase bile secretion. Increase bile secretion. And CCKPZ is produced by eye cells present in the duodenum and it causes contraction of GB. It also causes contraction of GB and relaxation of sphincter of Forty so that stored bile comes into the duodenum. And factors that stimulate CCKPZ, cholecystokinin, pancreatinine, hormone is fat or protein end products of digestion. This stimulates CCKPZ and when it is stimulated, it causes contraction of gallbladder and relaxation of sphincter of body so that stored bile comes into the duodenum. This is about regulation of bile acid synthesis. Two terms are there, cholagogues. Cholagogues are substances that stimulates gallbladder contraction. This is known as cholagogue. Okay, thank you very much for watching. Please like and share this video. And if you have any doubt, please comment. I will try to answer your or clear your doubt. Thank you very much.